This is a production of Cornell University.
Yeah, so as Chase mentioned, my work focuses mainly on shrub willow, and specifically Salix purpurea. And I'm really interested in sex determination and sort of the genes and mechanisms that are involved in this process. So a little bit of background on shrub willow. Willow belongs to the genus Salix, and this contains over 300 species. And among these are the shrub willow or section Vetrix. And these are a short rotation coppice bioenergy crop. So they're grown in high density plantings such as this, where they can be chipped coppice and chipped every 2 to 4 years. And then those chips can be burned to generate heat and electricity. Salix is interesting in that it's dioecious, so only about 5% of angiosperms are dioecious. They have separate female and male plants and with very different floral morphologies. And my research focuses on the species Salix purpurea as a model system for the shrub willows and more generally for the Salicaceae family, which also includes the genus Populus or the poplars, which are another important bioenergy crop. So I realize a lot of you have a background in plants, so you might not know a lot about sex determination. So there are two types of heterogametic systems that can be found in angiosperms. So the first is the ZW system, or female heterogametic system. And this is where females are ZW and males are ZZ. So the W chromosome is only inherited through females. So you might be familiar with this as the system that's present in birds. Kiwi fruit is an example of a crop species that has a ZW system. In an XY system, in contrast, the males are heterogametic. So males are XY, females are XX, and Y is inherited through the male line. And so this is probably the one you're familiar with in mammals, including humans. Also, papaya and asparagus are some crops that have this system as well. So Salix is really interesting because we see both XY and ZW systems in this genus. And so one way that this figure here is a little misleading is that this shows sex chromosomes being very different or heteromorphic. But in Salix, these sex chromosomes are actually homomorphic. So they can't really be distinguished through karyotyping. Much of the sequence is conserved. And the sex determination region is actually a section of the chromosome, and the rest is just an autosome and can be treated like any other chromosome. And in Salix purpurea, the SDR is on chromosome 15. It's a ZW system, and it's about 6.8 megabases, and this has about 400 genes in it. So with over 400 genes in this low recombination region, that presents a challenge for trying to parse out which genes are actually involved in sex determination and which are just linked. So this is a tree generated by some of our collaborators at West Virginia University and Texas Tech University, summarizing the different sex determination systems across both Populus and Salix. So Salix purpurea, as I mentioned, is a chromosome 15 ZW system. And many of the other shrub willows also share a chromosome 15 ZW system. However, as we get to some of the more basal Salix, it changes a little bit. So Salix exigua actually has a SDR on chromosome 15, but it's actually XY. And then in Salix nigra, it's a chromosome 7 XY system. And then in the poplars, we observe chromosome 19 XY, chromosome 19 ZW, and chromosome 14 XY systems. So this is very fascinating as it indicates that throughout the evolution of the Salicaceae family, which again, as I mentioned, there's over 300 species of willow and about 30 species of poplar, all of which are dioecious. Throughout the evolution of this family, the sex determination region has both shifted chromosomes and switched heterogametic systems multiple times. And so this really presents Salicaceae and Salix in particular as a very useful system to study sex determination in angiosperms. So as I mentioned, my interest is really in determining what these master regulator genes are of sex and sort of what are the pathways that they regulate sex dimorphism. So with the help of JGI, we obtained catkin RNA-seq data from 90 males and 90 females in an F2 family. And this F2 family was actually generated from our reference male and reference female. So we have reference genomes that we were able to pretty readily apply to analyze this data set. So this is a summary of the bioinformatic pipeline I used, and I won't go into too much detail about this right now, but if you have questions, I'd love to answer them. But basically, we did differential expression analysis, we did network analysis, we incorporated both RNA-seq data as well as GBS SNP data to do EQTL mapping. And we also had some small RNA data from a subset of these samples that we were also able to map and perform differential expression. So this is an MA plot. So the x-axis is the total overall expression of the of the genes, and the y-axis is the log to male to female fold change. So positive values are more male differentially expressed and negative values are more female differentially expressed. And every red dot is a significant gene. So right away you can see that there are thousands of genes that are differentially expressed. These are located all over the genome. And there's actually slightly more male differentially expressed genes than female differentially expressed genes, which is also quite interesting. But as I mentioned earlier, I really want to know, okay, what are the master regulator genes that are controlling all of this? Because the leading model of sex determination sort of posits that you would only need one or two genes in the sex determination region to do this. In this case, we're looking for a gene that would confer femaleness and then a gene that would also suppress maleness in the females to get different sexes. So we sought out to identify candidate genes. And so to do this, we used network analysis was one of the methods. And so this shows modules that were developed through that network analysis, and green bars indicate more female correlated and red are more male correlated. So there's several modules that emerge that are highly female and highly male correlated, likely representing the female and male sex dimorphism pathways respectively. So when we were looking at candidate genes, we first wanted to see that they were unique to the W SDR, so present in 15W and present in females, but also absent from males and absent from Z. And then we looked at genes that had module membership, particularly in this TAN module, which has a 0.85 correlation with females. And then we wanted genes that, of course, had significant and substantial differential expression. And lastly, these genes had to have a functional annotation that was consistent with something that might cause sex dimorphism. So hormone signaling, transcription regulation, transcription factors, things that could actually reasonably regulate all the dimorphism we see. So using these different criteria, we developed a short list of 8 genes from 5 different gene families. This is a substantial improvement from the over 400 genes that are in the SDR. And so we also have expression data that was included in the same data set from an unrelated monoecious plant. So this monoecious plant has both male and female floral tissue. And so we could use expression on this to hypothesize whether one of our candidate genes was a male suppressor gene or a female inducer gene. And so among these are 4 copies of an Arabidopsis response regulator, which is a negative regulator of cytokinin. And this is interesting because these are thought to be the master regulators in poplar. And they're also showing up as being strong candidates in willow as well. However, in poplar, they're thought to have both a female promoting and a male suppressing role. But in willow, we think that if they are master regulators, they probably only have a male suppressing role because we see very high expression in females, but we see very low to no expression in monoecious and male individuals. There's also this DRB1 and Argonaute4 genes. So these are both involved in transcription regulation through RNA silencing and RNA and DNA methylation. And these are located adjacent to these ARR17 genes. And they're also, based on their relative monoecious to female to male expression, are thought to be male suppressor genes as well. And then additionally, there is a zinc finger transcription factor, which isn't really characterized, but also has expression that would implicate it as being a possible male suppressor gene. And then we actually have one gene that we think might be a female inducer gene, and this is the GATA15 transcription factor. So this is involved in shoot and floral development and morphogenesis. So that alone gives it presents it as a very strong candidate for being involved in sex dimorphism. And furthermore, we see very high expression of this in both females and our monoecious plant, but little to no expression in males, which indicates it might be a female inducer. And this was also in previous research was actually found to be differentially expressed very early on in shoot differentiation when catechins were just starting to develop, which indicates it might be one of the earliest cues in sex dimorphism development. So from there, I sort of sought to identify what are some of these intermediate genes, right? So we have some candidates from master regulator genes. We know the thousands of genes across the genome that are differentially expressed, but what are sort of the intermediates between these that are directly regulated by the SDR and then in turn regulate these thousands of genes across the genome? So I use EQTL analysis to begin to answer this question. And EQTL analysis, or expression QTL, basically associates the expression levels of genes with genomic polymorphisms. So we found several thousand genes that were associated with the SDR, but then we narrowed them down to genes that were actually had relevant functional annotations. So we identified 54 genes that had associations with the SDR, but were located in other parts of the genome that were involved in either floral development or secondary metabolism. And that's because these are the two primary sex dimorphic traits that you can observe. So if you look at the floral tissue, they obviously have very different morphologies, and then their secondary metabolite profile, the terpenes and the phenylpropanoid pathway genes or compounds are very different. So we found multiple MADS box and floral development genes that were differentially expressed and associated with the SDR, as well as many phenylpropanoid and terpenoid pathway genes. And this figure here, which I outlined in red, so these are all many of the flowering pathway genes and sort of how they interact. I outlined in red the ones that we actually found have EQTL with the SDR. So these could represent entry points for the SDR to regulate floral development and in turn whether male or female flowers develop. We were also interested in transcription factors, so these can control the expression of many genes simultaneously. They're very strong candidates for being these intermediate genes that could be responsible for this widespread sex dimorphism and differential expression. And so among these, we found multiple transcription factors also involved in secondary metabolite biosynthesis in floral development, as well as gibberellin signaling as well. So we have this list of both candidate genes and candidate intermediate genes, but we really need to do functional annotation or functional analysis. So we'd like to, and this will help us to actually determine, okay, do these genes actually have an effect on sex dimorphism or floral development? How do they have that? And then what are some of the pathways by which they're actually involved in this? So in the future, we're hoping to do DAP-seq, ChIP-seq, and bisulfite sequencing on some of the transcription factors, as well as protein-protein interaction on some of the non-transcription factors to further characterize their genes they associate with. And we'd also like to do functional knockout and overexpression as well. And so with that, I'd like to thank the current and former members of the SMART Lab, our collaborators across both the US and China, and the NSF grant that has supported this research. And with that, I'll open it up to questions.
Great, thank you, Brennan. So everyone remember, submit questions to the Q&A tab. I'll start with a question because I'm a fellow willow researcher. So I believe, and correct me if I'm wrong, there's some evidence to show that there's a higher yield phenotype for females, is that correct?
Yeah, so a previous member of the SMART Lab, Fred Broker, actually did some research on this, and they found that there was some quantitative yield biomass traits, such as crown diameter, that are sexually dimorphic. However, so, they are quantitative traits, so they can't, you can't go out to the field and if they're not flowering, tell what's a male or female.
Is there anything in your differential expression data or your WGCNA analysis that might suggest a hint towards the mechanisms for why those are happening? Why the crown is a little bit larger, any of that?
Yeah, no, it's not apparent. So these RNA-seq data were collected from catkin tissue, so floral tissue. So it would be difficult, right? So when you're talking about biomass traits, you're often talking about the stems or the leaves, and that's not where our tissue's from. So yeah, it'd be hard to answer that question with the data set we have.
Yeah, totally. If anyone has any questions, now is the time. And
just to discuss that further is, it's very likely that these genes are associated with sex, but they might not be actually, or those quantitative traits. They could just be linked. So it may a gene in the SDR that's not actually involved in sex dimorphism, but is linked to the master regulator genes that might be responsible for that quantitative trait dimorphism that has been observed.
This has been a production of Cornell University, on the web at cornell.edu.